Hi, this video is about the composition of the plasma membrane. We learned about the plasma membrane when we were studying the cell and its organelles, but we didn't say a whole lot about it. That's about to change. The plasma membrane has three really critical general functions in the workings of the cell. One is it creates a boundary between the inside and the outside environment of the cell. No boundary, no difference between inside and outside, no life. The second thing that it does is it allows for cell-to-cell communication. So cells can communicate with each other without having to share the inside of their cellular environment, because the two plasma membranes can communicate either by touching molecules or by sending molecules which bind to molecules on another cell for long distance communication. You probably have heard of hormones, and that's what's going on there. The third thing that the plasma membrane is responsible for is transport. That means that it's controlling what goes into and out of the cell. If it didn't control it, then there wouldn't be much point in having a boundary in the first place. Because it controls what goes inside and out, or goes in and out of the cell, we say that the plasma membrane is selectively permeable. Things can go in and out of the cell or across the cell membrane, but the membrane is selective about it. It chooses which things are allowed to go and which things are not. Now, I'm going to take a piece of this plasma membrane here that you see circled at the top and magnify it into this drawing. And then we're going to talk about the composition of the membrane. So membranes are made of three major organic molecules: lipids, proteins, and carbohydrates. Lipids account for 98% of the volume of the plasma membrane, whereas proteins account for the remaining 2%. Carbohydrates are really so light and so small by comparison that they account for less than 1% of the membrane composition. However, by volume, the proteins are actually much more significant because they have 50% of the weight of the membrane. Proteins are much more dense and heavy than lipids are. Now, of the lipids, there are two lipids that make up the plasma membrane. One is phospholipids and the other is cholesterol. You'll recall that phospholipids are made up of a phosphate head group, which is represented by this circle, and two squiggly fatty acid tails, which are hydrophobic, and the phosphate group is hydrophilic. So because of the amphiphilic nature of this molecule, it naturally arranges itself so that the fatty acid tails are touching each other and the phosphate groups are on the outside. And it creates a bilayer. It creates a bilayer sphere naturally by itself without any intervention. In this bilayer, the phosphate groups are facing the outside and the inside of the cell, whereas the fatty acid tails are pointed to the interior where there is no water. Therefore, the phosphate groups are happy because they are hydrophilic and they are next to water. And the fatty acid tails are also happy because they are hydrophobic and they don't have to interact with water at all. In addition to phospholipids, we have cholesterol, which is represented by this ring structure. Cholesterol plays an important role in the plasma membrane because it allows for membrane fluidity, or for the membrane to be able to move without rupturing. Okay. The proteins in this diagram are these two red blobs, and there are many more proteins than this, of course, but this allows me to show you the two categories that proteins fall into that are in the plasma membrane. If the protein goes all the way through the phospholipid bilayer, we call it a transmembrane protein. If the protein is entirely on one side of the membrane, we say that it is a peripheral protein. Both of these proteins are usually associated, and I'll describe a situation where they work together in just a moment. Regardless of whether the protein is transmembrane or peripheral, it is associated with the cytoskeleton. The cytoskeleton allows the cell to put a membrane protein exactly where it wants it and keep it there, or move the membrane protein to another place. This is particularly important when you think about cells that have two different functions depending on which side of the cell you're on. In this type of cell, which we can imagine is a cell in the small intestine, one side of the cell faces the nutrients that are in the small intestine, and the other side of the cell faces the bloodstream. So the plasma membrane that faces the nutrients needs one set of equipment in order to move those nutrients into the cell. And the part of the plasma membrane that faces the bloodstream needs another set of equipment to move those nutrients out of the cell and into the bloodstream. Okay, so I talked about the cytoskeleton, all of these. Oh, one other thing. When we draw a membrane just by itself like this, not part of the whole cell, just a section of the membrane, by convention, the top part is the outside of the cell and the bottom part is the inside. I will usually write this when I'm making diagrams, but you may find that there's some diagrams in textbooks or online that don't provide that, and they still assume that you know that this is the outside of the cell and this is the inside. Before I go into the different kinds of proteins, I want to say something very quickly about the carbohydrates, and then we can forget about them. The carbohydrates do not exist in the plasma membrane by themselves. They exist connected either to the phospholipids or to the membrane proteins. Because the lipid and the protein are the dominant molecules and the carbohydrates are just extras that are attached, we refer to them as glycolipids and glycoproteins, which means that there's a little bit of sugar, glyco, on this lipid, or a little bit of sugar on this protein. And these do play an important role. The carbohydrates that are on the lipids, the glycolipids, those form a glycocalyx or a protective layer that's important to protect the cell membrane from the external environment. For instance, this one, which is facing the small intestine. So the glycocalyx helps to protect the surface of the cell from any damaging materials in the external environment. The glycoproteins, the glycoproteins act as cell ID molecules because each one of us has a unique conformation of this glycoprotein. This is what allows our immune system to say whether a cell belongs in our body or does not belong in our body. And it's also critical when it comes to doing blood transfusions and organ transplants. Okay. So let me erase a little bit of this and let's talk about the different kinds of proteins that we find in the plasma membrane. Okay, so get rid of this and get rid of this. This is what we're talking about now. So the first type of protein that we find in the plasma membrane is one for cell identity. And that's pretty much what I've drawn right here. This particular protein is a glycoprotein, and this arrangement of the carbohydrates on this protein is unique to this individual and allows the immune system to know whether this is self or this is something foreign. Now, other proteins in the plasma membrane may help to hold tissues together. So for instance, if this membrane protein has a particular part that it has high affinity to another protein on another membrane, they will grab together kind of like Velcro and keep those two cells together. If we didn't have cell adhesion molecules or things to hold the cells together, we could not have tissues, at least not the tissues that form layers or clusters. All right, so we have cell identity, cell adhesion. Now I want to talk about an example of a plasma membrane protein acting as a receptor. Okay, so here's our membrane protein. Here's part of it that sticks out, And that part of the protein is specific to a particular hormone or signal. So this would be a hormone, for instance, or more generally, these are referred to as ligands. All right, a ligand, to ligate means to tie together. So a ligand is something that ties together or binds with its specific receptor on the cell. Okay, so when it binds to this protein, it's going to cause the protein to change its shape simply because of the chemical reaction between the charges and the hydrophobicity of the amino acids here. And when this protein changes its shape, it causes a change in the peripheral protein next to it. So the peripheral protein will dissociate or leave the transmembrane protein, and it will go into the cell where it will cause a number of different effects. It could go to the nucleus and cause the nucleus to open up a gene so that we can make a new kind of protein. It could activate enzymes inside the cell. Really, the number of things that it could do is enormous. I'm just going to say that it causes a cell response. So this is what we were talking about when we were saying that membrane proteins allow for cell communication, or the membrane allows for cell communication, because a cell that's further away in the body that produces this hormone, once the hormone connects to its receptor in the plasma membrane, this cell will respond. So it received the communication and responded, and this allows cells in our body to work in a coordinated fashion. Okay, so that's receptors. Sometimes plasma membrane proteins work as enzymes. So for instance, in the small intestine, there are enzymes that are floating around with everything that's being digested, but there are also enzymes that we find embedded in the plasma membrane. And these enzymes, just like all enzymes, are specific. Oh, I shouldn't make that a triangle, should I? Because I just used triangles. We'll make it a different shape. There. All right, so enzymes are specific to their substrates. Every enzyme is. So floating along in the small intestine comes a substrate. Let's say it's a disaccharide that needs to be converted into monosaccharides. So this substrate will bind to the enzyme, and the enzyme will cut it in two. And then the two parts of the two products, terrible drawing, but the two products are then released, where they can be taken up by another type of protein in the cell membrane. So we have enzymes that are embedded in the membrane as well. Finally, we need some way to get these products, which I did not label, to get these products into the cell, or to be able to get something out of the cell that the cell does not want or need. And this is where we're going to talk about transport proteins. Now, for this one, I'm going to use a different drawing of the plasma membrane, and I'm not going to spend time drawing all of these phospholipids. So instead of drawing all of this, I'm now going to draw the plasma membrane as two lines, and in those two lines, I will have plasma membrane proteins, okay? All right. Transport proteins come in two major varieties. The first is called a channel. A channel is a transmembrane protein that sits in the membrane and allows substances to move either from the outside to the inside of the cell or the inside to the outside of the cell. So this is a channel. Now channels, if you could make yourself small enough to be in the cell or outside of the cell and look through, you would see that the channel is open between the outside and the inside environment. You would actually be able to see to the outside of the cell if you were inside. That doesn't mean that they're always closed, however. Channels often have a gate or a part of the protein that can swing away from the opening or swing towards the opening and close the channel. What opens this gate can either be a ligand, or it can be a change in voltage inside the cell. Voltage is a way of talking about whether it is becoming more positive or more negative. Or it can be operated mechanically, meaning that if something stretches or pushes, it will cause this channel to open up. This is particularly true in our inner ear, where movement opens channels, and that's what sends the signal to the brain so that we know where we are. Okay, so channels are open, open continuously between the inside and the outside, though they may have a gate. So there is an opening there. I know I'm saying this over and over again because I'm making a comparison. Channels are also incredibly fast because all that's required is for the channel to open, and substances can move according to diffusion, which we're going to talk about shortly. So channels are capable of moving about 1 million molecules per second. You do not have to know that number. I just want you to appreciate how fast these channels are in terms of allowing substances to move. Channels are also what we call selective. That means that because of the size of the pore and also the charge of the amino acids inside the channel is selective and will strongly favor only certain ions moving across. So for instance, we have sodium channels, potassium channels, chloride channels, we even have water channels. And each one of them is able to select that particular type of molecule because of the combination of how wide the opening is in the channel and the charges inside the channel itself, the amino acid charges. All right, that's channels. Now, the other type of transport protein is called a carrier or a transporter. Now, these are different from channels because carriers and transporters do not have an opening between the outside and the inside of the cell. The transporters and carriers are only able to have an opening facing either the outside or the inside at any particular moment. And there are lots of ways to draw them, but the simplest is this. Okay, so they sit in the plasma membrane, and carriers are specific. Well, first let's say that they are not open to both sides. And they are, they require movement. So the protein itself has to change shape, and therefore they are slower. So they can allow between 10 and 100,000 molecules per second to move through, which honestly is still really impressive, but it's not nearly as impressive as 1 million per second. Finally, comparing to channels, carriers are specific as opposed to selective. And that is because they have a site which only fits a particular substance, and only that substance can fit in that little pocket. Specific, just like we saw with enzymes and with receptors. Once this substance binds to the pocket, it causes the protein to change its shape. It's called a conformational change. And it changes its shape so that now the opening is facing the other side of the cell, which allows our substance to leave the transporter and be moved across the membrane. So the transporter sits like this, something binds, changes its shape, and now that substance is released. Now, I've shown this moving substances into the cell. The channel can have substances moving into the cell, or substances can move out of the cell. Same thing here. So the substance that just got moved into the cell, the whole thing could operate backwards. And the carrier protein simply moves back and forth between these two positions to move the substances that it carries. Now, you will hear these transporters discussed in terms of how many things they carry and in which direction. So what I've drawn here would be referred to as a uniporter because it transports one substance from one side of the cell to the other. Sometimes a transporter will have two pockets in here and can transport two substances from one side of the cell to the other. And that's called a co-transporter. They're both moving in the same direction. And sometimes transporters will move one substance in one direction and will move another substance in the opposite direction. And those are called antiporters or exchangers. Okay, so we've got channels and we've got carriers and transporters. We're going to talk about the law of diffusion and osmosis and volume changes in the cell in my next video. But for now, I just want to introduce one other special type of carrier that's going to become important later. All right, so it also has specific sites for whatever it is transporting across. I should have drawn it open somewhere. There we go. But what is different about this transporter, and you will see all of them notated this way, is that it requires ATP in order to operate. So artists will draw this and we'll write ATP in here. And what that means is that this particular carrier, in order to move the substances that it is specific for moving, has to use a molecule of ATP. So if this one is moving circles, right, the circle can bind to its pocket on the transporter, but the transporter is not going to do anything until a molecule of ATP comes by and drops off some energy. All right, so ATP gives the transporter its energy, and then it's able to make that conformational change. We say that this particular kind of transporter is called a pump. And you will soon see just how important pumps are to the function in our body. Until then.